in today's session, we'll be discussing about the questions that are related to Sanger sequencing. Okay. So Sanger sequencing, it is the first method to sequence or to find out the sequence of any unknown piece of DNA. Okay. Yeah. So pentose sugar, nitrogenous base, and phosphate group, right? All these together are called as nucleotides. Okay, and without phosphate it is called as nucleosides. Okay, so DNA is made of these nucleotides chains. Okay, so this unit of DNA is nucleotides. So for the DNA polymerase, for replication to happen, these nucleotides are also, we can call it as DNTPs. Okay, so these DNTPs are the substrate for the replication to takes place. Okay, so every DNA, this is a pentose sugar. Here, nitrogenous base is attached. At this point, we have the phosphate group, right? Phosphate group. Okay, and in this phosphate sugar structure, we have hydroxy groups here, which is present. 1, 2, 3, 4, the fifth carbon. So yeah, so this is the numbering. So in 2 and 3 we have the hydroxy group present, and if at the second position the hydroxy group is not present, then this is called the deoxy sugar, right? Deoxy sugar. So this is the deoxynucleotide phosphate which is present in DNA. Okay, and this 3' OH group which is involved in the phosphodiester bond formation with the next nucleotide. Okay, so this group is involved in the phosphodiester bond formation, okay, with the next group, with the next nucleotide, and that's how the chain continues. Okay, so this is how it happens. So in DNA, if we don't have this 3' hydroxyl group, then the next nucleotide cannot join with this at this particular residue. Okay, so that is the modification that has been used in the Sanger sequencing. Okay. So, so they use a dideoxy. So here also phosphate group is not there at second, as the third position of the sugar. At both the places hydroxy group is not present, only H is present. So this is called dideoxy nucleotide triphosphate. So this is the dideoxy. At both the places that hydroxy group is not present. So if this hydroxy group is absent, the DNA polymerization will not take place. Okay, like if there is a substrate, like this is your substrate, and for DNA polymerization or DNA recombination, we need a primer, right? A primer that will bind. A primer that will bind here, and the next nucleotide will come and attach here, and then another nucleotide, and then another nucleotide, and they keep on they keep on synthesizing the phosphodiester bond in between them, and that's how the chain will elongate. Okay, but if the 3' group doesn't have the hydroxy uh, group in that, or the hydroxy group is absent, then the elongation will stop. So as I'm saying that in the if we use dideoxynucleotide triphosphate to synthesize the DNA replication will stop. So in Sanger method, what they do, okay. So there also they perform a PCR to find out the sequence of an unknown template. Okay. So if you have a sequence, like suppose if you have a particular sequence, like suppose unknown DNA is there, then so they use the dideoxynucleotide triphosphate. So they use four different types, like the G dideoxy uh, guanosine triphosphate, deoxyadenosine triphosphate, dideoxycytosine triphosphate, and deoxythymidine triphosphate. All those four they use. So if in PCR, what they provide, they make four different tubes. They use the same template to amplify the particular piece of DNA, but there they provide primer, they provide all sets of DNTP, all mixture, and one particular dideoxynucleotide triphosphate. So in the they have four tubes, uh, and in all the primer, DNTP's template, everything remains same, but dideoxy, one particular dideoxy sugar they provide. Sorry, I unable to do with the laptop marker, so I'm just going to explain just like that. Please try to cope up. Okay, so dideoxy G, dideoxy ATP. So if there is a sequence, if this is the sequence, okay, and this you want to amplify, okay. So in this particular tube, what will happen, your primer will anneal, and as all the DNTPs are present, okay, but only this one is the dideoxy one. So here the DNA will synthesize, but wherever this one needs to incorporate, if so here, if in the is particular nucleotide sequence where this one will go and incorporate, okay, so this will be a random uh, integration of this dideoxy, because DNTPs are provided in abundance, but this one in a very small concentration. So all the other nucleotides will go and find do the hydrogen bonding with the complementary bases. So suppose as this position the C is present, so this particular one will go and make the hydrogen bonding, but as the dideoxy one is there, the 3' end doesn't contain a hydroxy group, so elongation of this particular chain will not take place. Okay, so this fragment will stop here. So you will find a fragment, this particular fragment, DNA fragment. Another scenario, suppose here normal DNTP will go and attach, then again synthesis will continue, and here again this particular fragment will form again. Okay, and then again next one, if here normal DNTP will go and here dideoxy one will bind, then yeah, so it was just like that to find out the sequence of the unknown DNA, they set up a reaction using four different tubes where they give you the where the template, unknown template is given, for which you find out the sequence. Primer will be there, DNA polymerase will be there, DNTPs will be there, and very less concentration of on any one particular dideoxy triphosphate. So suppose in one tube all will be there, but only dideoxy guanosine triphosphate, another tube, another one, another tube, another one. So like this 4 tubes will be made. And if the unknown sequence is this, I have given this particular sequence, and here if DNTPs will go and do the complementary base pairing, but if at any place the dideoxy one will go and bind, then the elongation of that chain will not happen, it will stop right there. So suppose here the dideoxy guanosine triphosphate is being attached, then only this short sequence will synthesize. If the deoxyguanosine triphosphate will attach, then the sequence will continue, and the in the next place maybe the dideoxy one will go and bind. So you will get varied length of the DNA pieces that will. So here accordingly through the in the another tube also you'll find some other sequence. Okay, and these will be radio labeled. Okay, so when all particular tubes, like suppose in 4 lanes, if you run the gel like this, one this tube, then the dideoxy adenosine, this and this. Okay, so if you are getting a band pattern like this, okay, if this is the band pattern you are given, so according to size, this is the small size, this is here the larger fragment will come, and the readout you will do from the bottom, because that will be the first nucleotide that is present in the DNA. That is why the shortest fragment is the synthesized. So here if we'll read out the sequence, that will be in the order of, so here the first sequence is formed in this lane, that means the sequence is C, and then another sequence is also there in the same lane, and then after that a band is being formed a slightly above compared to this one, then it will be A, and then G, and then T, T, A and G. So this will be the readout after running all the samples. Okay, and the sequence will be in this direction, 5 prime to 3 prime. Okay, so this will be the readout at which particular sequence the chain terminated. Okay, so this is the complementary base pairing, complementary strand sequence. So the original strand sequence will be complementary to this in 5 prime to 3 prime direction in this way. Okay, so that is the readout. So let's do the question. So here the question is, an oligonucleotide was sequenced by the dideoxy method of Sanger and the following autoradiograms was obtained. Okay, so these are the different lanes where in each lane they have provided, suppose in this lane they have provided dideoxy adenosine triphosphate, in this one the T, C and G. Okay, and this is the band pattern they have got. So seeing this, if we have to find out the sequence or we have to deduce the sequence, so let's write this sequence: G, T, C, C, T, G, T, A, C, A. Okay, so this will be in which direction? This is 5 prime to 3 prime direction. Yeah, so this is the sequence of the oligonucleotide. So is this option given? B option, right? Yeah, this is the same. GTCC, TGT, ACA in 5 prime to 3 prime. There are, so these A and B both have the same sequences. You have to very carefully see the 5 prime or 3 prime and where is which. Okay, so seeing this you can identify the same sequence is there, but 3 prime to 5 prime, but original sequence from this you'll deduce in the odd sequence of 5 prime to 3 prime in this direction. So this is the correct option.
And this is my NPTEL reference. Thank you.